Thank you. Thank you all for being here today. I think this is on. Yeah, it's on. Okay, so my thesis proposal is on genomic instability of solid tumors and clinical applications. So genomic instability is a hallmark of all cancer types. So basically this includes high frequency of mutations and chromosomal instability. And so basically what this figure is showing is chromosomes that are located in the nucleus of a cell, and then each section is a gene. And then below that is a human karyotype, which is the original way that chromosomes were viewed under a microscope. And you can see that each chromosome has kind of a different shape and structure. And so this basically exists on a scale of genomic variation. So clinical cytogenetics includes discussions about gene fusions and chromosomal instability on a much larger scale. And so you can see that on the right side of this scale. This can include whole chromosomes or parts of chromosomes that are duplicated or deleted, whereas genomic instability also includes point mutations on a smaller scale, which is on the left side of this scale. And so this is basically like a one nucleotide change, whereas chromosome abnormality is on a larger scale. So researchers have used information from different scales to summarize instability in solid tumors, and the clinical utility is uncertain, whereas chromosome abnormalities such as these structural changes or gene fusions are routinely used for clinical decision making. So when we talk about clinical decision making, this really depends on the cancer type, and this can include different types of genetic variation, which is useful for clinical decision making. So one of the most known examples is the Philadelphia chromosome in CML. And so basically what this is a reciprocal translocation from chromosome 9 to chromosome 22. And what this does is it creates a fusion gene known as BCR-ABL1, and this causes uncontrollable cell growth in CML. And this is used for clinical diagnosis, predicting survival, and treatment options. And so basically, a cytoband tells you where on the chromosome you are. So in this case, we have Q34.1. So basically, the cytoband is 9Q34.1. Another example is in AML, which also uses cytogenetics for clinical decision making. And so the example for that is PML-RA-RA, which is also a fusion gene. And this is a translocation from chromosome 15 to 17. And this is used for diagnosis of subtypes of AML and also for treatment plans. But when we think of like breast cancer and ovarian cancer, it's mostly subtyping based on mutations. And so that's more on the smaller scale that I was talking about. And so an example of this is like BRCA1 and 2 mutation, which is used for diagnosis and treatment plans. So there are different types of chromosomal abnormality structures. This includes deletion, duplication, inversion, substitution, and translocation, as we saw in the previous example. And so there are basically lots of different ways to measure this chromosomal abnormality structure, and there's currently no good way to summarize this. So in hematologic malignancies versus solid tumors, basically, recurrent findings have been consistently associated with clinical measures in leukemias, and the samples are found in the bone marrow. And the chromosomal instability measures are found in almost all the leukemia cells, whereas in solid tumors, the findings are not as consistent compared to leukemias. And basically, the quality of the chromosomes are suboptimal because of the cancer cells before culturing. And there's also complex chromosomal changes due to the tumor progression. And so it's difficult to identify primary chromosomal changes for each tumor type. However, there are new techniques and better characterization of solid tumors, which has made chromosomal instability a promising marker for solid tumors. So again, despite these challenges, chromosomal abnormality has had clinical advances in research, such as in breast cancer. They found that the FISH assay, in combination with HER2 and chromosome 17 sequences, this was more accurate in, than protein expression alone in selecting patients that would respond to targeted therapy. And in head and neck cancer, the FISH assay was also used to monitor surgical margins by identifying chromosomal imbalances in the head and neck tumor samples. So basically, chromosomal instability scores were developed to measure different kinds of instability because these methods are not routinely used in clinical decision-making for solid tumors. So I'll be talking about 3 different chromosomal instability scores, and so this incorporates gene expression, copy number, and somatic mutation. So the first one I'm going to talk about is the CIN25, which is basically a 25 gene signature across patient samples, and this is used as a classifier for prognosis. And so basically, these 25 genes were selected based on the correlation with functional aneuploidy, and this is calculated as a T statistic across diverse tumor types. And so on the left, you can see the heat map, which is basically each row is the gene from the 25 gene signature, and each column is the patient. And the net CIN signature is basically the net score for each patient. And then the middle line is the average. And so they classified each patient into low and high categories based on the average. And so as you can see from the survival plot, the patients with the lower CIN score actually survived more than the ones with the higher score. And so basically what this survival curve is a Kaplan-Meier survival curve with a p-value at the 0.05 level. And this calculates the fraction of patients known to have survived across time. And the p-value corresponds to a log-rank test comparing the pair of survival curves. And so you can see that the patients that were classified with a lower CIN score, they actually survived, a higher fraction of the patients survived than the patients with a higher CIN score. And so this CIN25 performed well for 12 out of the 18 data sets that they tested. And in this particular example, this is the breast cancer data set. So I'll move on to the next score, which is based on both copy number and somatic mutation. And this is basically a weighted calculation of the copy number and the somatic mutations. And so what they found is that a higher GI score is associated with the higher survival rate in ovarian cancer. And this is regardless of BRCA1 and 2 mutations. And so basically they adjusted for the BRCA1 and 2 mutations by computing survival for high and low GI scores with each group containing the BRCA1 and 2 mutations versus BRCA wild type. And so they found that the higher group had a higher survival rate, meaning that this score can be used to further divide BRCA mutant ovarian tumors. And so this is kind of the opposite finding that they had for the CIN score, where a higher CIN score was associated with a lower survival rate. So then the last score I'm going to talk about is the C-index, which is a package on bioconductor, and this is based solely on copy number. And so they calculated this score for each chromosome and for each sample. And so they calculated this at the chromosome level and then also at the cytoband level, which is on a smaller scale. And they found that a higher loss CIN score was associated with better prognosis. And almost the entire chromosome 20 was lost in the relapse-free group. So this is a little bit different group because this is comparing patients that are relapse versus relapse-free, whereas the previous ones were based solely on survival. So this is the summary of the scores that I talked about. And so the CIN25 basically performed well for 12 out of the 18 data sets, and they used the gene expression data only. And their finding was that a higher CIN score was associated with a worse prognosis, and this was tested in multiple cancer types. And then the GI score was tested for one tumor type, and they used both copy number and somatic mutation. And a higher score was associated with better prognosis in the ovarian cancer data set. And then the C-index score was also performed well in one tumor type, and they used only copy number and a higher loss score, meaning that a higher deletion score of the chromosome was associated with a better prognosis for colorectal cancer. So basically, there's a gap in the research where associations with clinical outcomes have been inconsistent, and there's no consensus about the best way to summarize these scores. And recurrent cytogenetic abnormalities like we see in AML are not included in the scores for solid tumors. So there's a potential clinical value of CIN as a predictor of cancer progression. So the hypothesis is that using combinations of these current chromosomal instability measures, which utilizes different data types, this will provide a greater ability to predict outcomes across a wide variety of cancers. And so the motivation for this is that this will provide researchers a way to annotate the cancer samples with these different measures of genomic instability, and to be able to provide evidence for which measures are most predictive of clinical features in a variety of solid tumors. So this brings me to aim one, which is basically to develop an R software package to be launched through Bioconductor. And this will provide comprehensive annotations related to genomic instability for TCGA samples. And this will include coding in R, Linux, and Python. So this is just an overview of the number of samples in TCGA. And this is just to show that there is coverage for all of the data types available in TCGA for these cancer types. And there is some variability among the samples, but we do have coverage for the clinical data, copy number, somatic mutation, and gene expression. So I'll be studying these 4 cancer types because the first 3 are for validation, and the last one for kidney cancer, that's a new data set that hasn't been implemented with the CIN scores. And so basically I'll be downloading the TCGA data from the GDC data portal, and this will include gene expression, copy number, and somatic mutation, and the levels just indicate that this is open source data, and so it's not controlled access, and that means that the somatic mutations don't contain any germline mutations. And then I'll also be downloading the recurrent chromosome aberrations in the Middleman database, which is basically more of the structural changes. And so you can see an example of that on the bottom table here. So basically when I'm processing the CIN score, I'll be annotating this for all of the TCGA patients. So the CIN25 is the gene expression, GI is the somatic mutation plus copy number, and then the C-index is the copy number at the chromosome and the cytoband level. And then I'll also identify the high frequency chromosomal abnormalities. And so an example of what this looks like in the Middleman database is the structural variation will basically be the cytoband. So in the first row, it's 1q10, so chromosome 10 on the bigger arm at location 10. And in this example, it's in insertion or also substitution. And so basically what happens is part of the chromosome gets inserted into a different chromosome. And this occurs in 18% of the breast cancer cases in the Middleman database, and then 25% in ovarian cancer. And then the next 2 examples are derivative and addition, which is basically duplication. And a derivative is similar to the other measures that I talked about, which is like a combination of inversion and deletion. So in this case, it's a deletion of chromosome 16 and inverted onto chromosome 1. So the validation will basically be seeking to validate what the previous studies found. And so this will involve checking the CIN25 score and seeing if a higher score denotes a worse prognosis in breast cancer. And then for the GI score, if a higher score is a better prognosis in ovarian cancer. And for the C-index, if a higher loss score is a better prognosis in colorectal cancer. So this will be validation of the scores using different data sets, but it will be the same tumor types. And so the last thing in AIM-1 will basically be the exploratory data analysis of the scores. And so I'll correlate the different scores to see if they're providing any new information. And if they are highly correlated, then this will affect the regression analysis with possible multicollinearity. And I'll check if there's any statistical differences in the proportions in the clinical variables, such as the age, tumor stages, and primary versus metastatic tumors. And then also be checking the differences in the score distributions across the tumor types. And so this is for the EDA. So then for AIM-2, I'll actually be investigating the predictive ability of genomic instability annotations across these variety of cancer types. And this is just an example of like what the CIN25 score, like what the visualization will look like for that. And so it's that same idea with the heat map and then the low and the high scores and then what the survival curve looks like for those. So some of the analysis questions are, is there a CIN combination score that better predicts survival in diverse cancer types? And basically I'll be doing a multiple survival analysis for this and also investigate whether a combination of CIN scores better predicts survival in any one score alone. And also, is there a significant difference in the CIN scores of TCGA patients in different tumor stages or primary versus metastatic tumors? And so to answer this question, I'll do a logistic regression to determine if CIN scores are significantly different between these groups. And then similarly, I'll be investigating whether a combination of CIN scores better predicts survival than any one score alone. And then the last question I'll be looking at is to create a region-specific instability score. And so this will be looking at whether the important cytoband scores are those known that have recurrent abnormalities. And so I'll determine if there's chromosome-specific or cytoband-specific scores that are more predictive than instability scores based on the entire genome. And so this is my timeline for my thesis. So this summer, it will mostly be working on, or I'll be mostly working on research, data collection, and then the software engineering of the our bioconductor package. So this will involve downloading and parsing the data across the 4 cancer types that I mentioned, and then also downloading, parsing the Middleman database, and then annotating the CIN scores, and then validation, and then wrapping that up into an R package. And then fall term, I'll be working on more of the data analysis with the scores, and then also the data visualization piece, which will probably be through Shiny. And then I'll also be looking at the best way to summarize CIN scores and which ones predict better. And so, as I mentioned before, the deliverables will be in our bioconductor package. And so this will basically function to process data from any cancer type from TCGA and supports a variety of data types, so math files, text files, CSV, or Excel sheets. And then basically this will output the clean data with the CIN annotations. And then I'll also do a separate piece, which is the data visualization tool, which will show the results, including heat maps, survival curves for each CIN score, and how they perform across each of the cancer types. And then I'll make recommendations about the most appropriate way to summarize genomic instability, and then a summary of which data types are useful for each cancer type. So the innovation of this project is that it will provide evidence for which measures of genomic instability are most predictive in a variety of tumor types. And tumor stage hasn't been implemented as a clinical outcome in the previous methods that I described. And then I'll also be investigating the utility of incorporating information about the known recurrent chromosome abnormalities. So the next steps of this research might involve the beginning of more clinically based research based on how the CIN scores do for each tumor type. So the limitations of this study include variability among solid tumor samples between the previous studies and the TCGA, which may affect the validation step of the previous studies. And there's also variability of data coverage for each tumor type, which will affect the score annotations. So, for example, some patients may have a GI score but not a CIN score because they're missing gene expression. And also the sample size differences across tumor types will affect the distributions of the score. And so basically, the breast cancer data set will have a more narrow distribution of scores versus the other tumor types, because it has almost twice the amount of samples. And then these are my references. And then I would just like to thank my thesis advisory committee and DMICE faculty and staff and students and my friends and family. So I'll take questions now. Okay.
So what do you think it would take to get a clinician to kind of accept the score?
Based on the evidence, maybe it would give them something to think about, especially with like breast cancer and ovarian cancer, you typically think about the mutations, but maybe if you add in expression, that might convince them like this actually predicts survival better or worse, and it might actually show you, you might be able to subtype that based on the tumor stage, and so that might be able to give them an idea of like how they might stratify treatment, and so it would just basically start that conversation.
So yeah.
Yeah. Okay. Yeah. Thank you.
It's real interesting from a clinical perspective. Well, the question, actually, I was first going to ask you what happens if you, if in the A1 you don't validate the existing models, but then I thought, well, maybe that doesn't matter because you're going to come up with new models, although I suppose it would be interesting for the world to know that no one's ever looked at like TCGA data and kind of compared it with these existing models.
Yeah, that was one of my questions too. I was like, well, what if I don't validate the original scores? And that's just something I'll have to think about. So maybe combining the scores would validate the previous studies, which would be really interesting. Or in terms of validation, yeah, it's almost like proving that there is so much variability in the tumor types, and that it really depends on the tumor, and that maybe there is some other way to score these.
Because as I
Okay, yeah.
Because in certain diseases, what you know, oh, it's right? And then you can ask things, if you're doing all that EDA, you can ask things like, how different are my TCGA populations from the study population? The answer's going to be wildly different. The TCGA had a collection method which actually was very biased, all comers, type of thing. So if you have a very well-curated ovarian or breast data set, it's dramatically different than TCGA one. That's one of the things you look at right away. And that's actually an important finding. It scores better than whole in these different groups.
Mm-hmm.
For these reasons, it's actually a clue.
Yeah, that's a good point.
Instability important.
Yeah.
It's not.
Okay. I do think that's interesting from the patient perspective, too. Because typically, at least for breast cancer, it's all about the BRCA1 and 2 mutations. So if you were able to incorporate expression and copy number, that might help them understand it more, too.
Although expression.
Oh, okay, yeah. Okay, yeah. Okay.
For sure.
Yeah, they're all primary tumor samples before treatment. So, yeah. All right, thank you, guys.